Genetic diversity, the advantage of sex. So, sexual reproduction is actually very difficult and complicated. It takes a lot of work. You need two individuals to reproduce. You need to spend effort to attract a mate, to keep a mate. Sexually reproducing organisms tend to reproduce more slowly than asexually reproducing organisms. It would be a lot simpler just to split in half and have two of you than to go through all the process of sexual reproduction. However, there is an incredible advantage to sexual reproduction, and that is that you produce offspring that are different than you, different than your partner, different than some other offspring of you and your partner, which means that sexual reproduction increases the genetic diversity of a population. This promotes survival because if you have all sorts of different combinations of alleles in individuals in the population, if there is some sort of plague or something that has a genetically encoded susceptibility, somebody in that population ought to be able to survive it if everyone is different. If everyone is the same or identical, then something that kills off one organism of the population will kill off the entire population, and that's the end of that organism. So genetic diversity is caused or is allowed by differences in genes. And when we talk about genetics in the next chapter, we're going to talk particularly about alleles. You can think about an allele as a version of a gene. So if you're diploid and you have two copies of the gene for eye color, your father might have given you information that would let you for having blue eyes. Your mother might have given you information for having brown eyes. Those are both the genes for eye color, but they're two different versions, two different alleles, and have the potential to give you different colored eyes. There are 5 ways to create genetic diversity, either by making new alleles in the first place or by combining them in different combinations. First, alleles are produced by mutation. So sometime when DNA polymerase is replicating the DNA, it makes a mistake, it doesn't catch it, that mistake is propagated. Now you have a slightly different code in the DNA that might produce a slightly different protein with a slightly different outcome or physical characteristic. So mutation produces alleles. After mutation produces alleles, then crossing over happening during prophase 1 of meiosis, segregation and independent assortment happening during anaphase 1 of meiosis, and fertilization after production of the gametes will all start putting those alleles together in new combinations. And that matters because if you think about it, imagine the comparative usefulness of having long legs and asthma compared to having long legs and really great lungs. The second individual has the potential to be a star track runner. The first individual probably won't be. So we're going to go through each of these 5 ways that genetic diversity is increased one by one. First, mutations. Mutations, if you remember, are changes in the nucleotide sequence of the DNA. If that change is not corrected, but is instead then used as a template and passed on, you have new alleles, new DNA sequences, new versions of a gene. These DNA sequences may or may not produce slightly different proteins. If you have, for instance, a missense mutation, maybe you put in a leucine instead of an alanine, and maybe that causes the protein to behave a little bit differently, which can increase the genetic diversity. Remember, most mutations are going to be harmful. A few might be helpful. Next, we have crossing over. Crossing over and synapsis happen in prophase 1 of meiosis. This is where the homologous pairs of replicated chromosomes find each other, pair up, kind of flop their equivalent regions over each other, and exchange information. So that instead of having the chromosome 1 that you inherited from your father separate from the chromosome 1 that you inherited from your mother, you create chromatids that might have combinations of what you inherited from your father and what you inherited from your mother on chromosome 1. So you create these patchwork chromosomes. This is going to be a way to allow new combinations of genetic information. You're going to give to your child perhaps a combination of the chromosome 1 from your father and your mother instead of just what your mother had or just what your father had. Pictures might be easier than words. You can see that you have 2 homologous chromosomes here. They are going to pass back and forth a little bit of information so that all of the same genes are retained, but here this purple chromosome has a little bit of information that used to be on the green. This green has a little bit of what used to be on the purple. Notice now that each of these chromatids is different. There's one chromatid that only has the green information, one chromatid that has the small bit of the purple with the majority green, one chromatid that has the majority purple but a small bit of this green information, and one chromatid exclusively purple information. And if you can see from the key over here, there actually are different alleles encoded on the purple chromosome 1 versus the green chromosome 1. So you are making new combinations of alleles by crossing over. Then, when first your homologous pairs and then your chromatids separate during meiosis, crossing over, even in the absence of any other reassortment, produces genetically unique gametes, genetically unique haploid cells. When genes are very near to each other on a chromosome, like this yellow dot is very near to this yellow dot, those genes have a low chance of being separated by crossing over. The crossover would have to happen between those 2 dots. There's a very small area where that exchange could happen to separate the 2 dots, and if there's only a few ways to do something, it's less likely to get done. So very near genes are likely to be inherited together as a unit. We will talk about this as linkage when we get to the next chapter. However, alleles that are far apart from each other, the crossing over could happen anywhere between those alleles, and alleles that are far apart are very likely to be exchanged. So likely that it's unusual to find 2 traits, 2 alleles that are inherited as a unit instead of being inherited independently. The next contributor to genetic diversity is segregation. Segregation is the separation of the homologous chromosomes. Each parent has 2 of each type of chromosome. They have pairs of homologous chromosomes, but they can only give one of those 2 pieces of information to their offspring because during anaphase 1 of meiosis, those 2 pieces of information are separated from each other. So if you have a case where an individual has one chromosome with an allele for functional insulin and one chromosome with an allele for non-functional insulin, segregation means that that individual is going to give his offspring only the non-functional or the functional, not both of those informations together. Because of segregation, you can only give one of your 2 pieces of information for each trait to your child. Also, independent assortment, which we haven't spoken up much, means that you have the chance to combine the information that you've got from your mother or the information that you got with your father on one chromosome with the information that you got from either of your parents on a different chromosome. Once you've made your gametes, all of which are different from each other because of crossing over, segregation, and independent assortment, then you have to take one random gamete from the mother, one random gamete from the father, and put them together to make a haploid cell that grows up to be a haploid individual. We call this process fertilization. There are so many different ways you can put together your genetic information to make gametes. Once you randomly choose one of your multitudinous potential haploid combinations of your genetic information and randomly put that with only one possible one of all the many combinations that your partner could make, the possible combinations of information that two individuals together could put into their offspring is approximately infinite, meaning that two people or two of some other animal could have an infinite number of children, and no two of those children would be genetically identical. This means that there is a lot of opportunity for difference in the population, a lot of opportunity for genetic variation. Some of these variations might be more successful, better suited for their environment. Some of them might be less successful, less suited for their environment. But when the environment changes, probably some of those individuals who are less suited for the previous environment will be better suited for the new environment. So the species gets to continue. Moving on from variation, we're going to talk about briefly some errors in anaphase. We call these errors non-disjunction. So this is a long word, let's break it down. Junction is holding two things together, and specifically talking about the two chromatids that are held together at the centromere of a replicated chromosome. Disjunction means to take apart the junction, and this is what's supposed to happen during anaphase. You're supposed to either take apart the tetrad to separate the homologous chromosomes, or you're supposed to take apart the replicated chromosome to separate the chromatids. Non-disjunction then means you're not going to take apart those connected pieces that you're supposed to be taking apart. And non-disjunction leads to chromosome abnormalities, either too many or too few of just one particular chromosome in the body or in the cell. So non-disjunction is when either the homologous chromosomes don't separate during anaphase 1 of meiosis, or when the replicated chromosomes, the sister chromatids, don't separate during anaphase 2 of meiosis. This means that the gamete that's produced either has two copies of a particular chromosome, one copy of everything else, but two copies, say, of chromosome 5, or the other product of that division will have one copy of everything else, but 0 copies of chromosome 5. If you have fertilization with a normal gamete, you're adding one more copy of everything, including chromosome 5. The zygote, the fertilized egg that came from the cell that has the gamete that has 2 copies of a chromosome plus one more copy, will have 3 copies. And the fertilized egg that came from the cell that didn't get a copy of that particular chromosome now has had one copy joined to it by the incoming gamete, only has one copy, not the 2, the diploid number that it's supposed to have. Often, the fertilization that results from non-disjunction, there's something so much wrong with it that the cell dies, that the embryo dies before it's even implanted. But there are a few non-disjunction events that are survivable. So you can end up with a baby that's born that has too many or too few of a few specific chromosomes because of an error in separation at anaphase.